Hi, Bobcats. In this video, we're going to take a look at the nature of light and how light can interact with matter, because that type of interaction gave us all kinds of information about how electrons are arranged in an atom. So our first objective is to describe light as being part of the electromagnetic spectrum. We want to distinguish continuous from line spectra, and to examine how matter can emit light as well as absorb light under different circumstances. It's standard to describe light in two different ways. And I think when you put the two descriptions together, you get a more complete picture of what light is all about. Light is a portion of what we call the electromagnetic spectrum. Well, that is visible light's a portion. When we use the word light, I guess it really does mean kind of the whole electromagnetic spectrum. We can use what's called a wave description or a particle description. The wave description of light will use terms like frequency and wavelength and energy. The particle description will use terms like photon. A photon is an individual packet of light energy. And so in the particle description, we talk about photons and the energy that's associated with that photon. The entire electromagnetic spectrum runs from gamma rays all the way down to radio waves. The gamma rays end is the high end, the radio wave end is the low energy end. There's this very, very tiny piece of the spectrum that our eyes are sensitive to, and that's known as the visible spectrum. And if we can talk about the electromagnetic spectrum in terms of light, in terms of energy, but we can also talk about it in terms of frequency and wavelength. Frequency tracks with energy, so high energy means high frequency, but wavelength is inverse to that. A long wavelength means that it is a low energy, or a short wavelength means that it's a very high energy. On the previous slide, if you look at the portion of the spectrum that is visible, you'll notice that we have what's called a continuous spectrum. All of the colors of the rainbow are present, and as you spread that light out, like through a prism, one color is connected directly to the next. They just are a continuous spectrum. Well, we can also get light from various elements and other substances by passing a lot of energy through a sample, and that causes the sample to emit light. This is basically how a neon lamp works. A neon lamp is a glass tube that has been evacuated, so that means all of the contents, all of the air inside of the tube has been pulled out by a vacuum pump, and then some neon has been backfilled into that tube. And if you put a really big electrical voltage across that tube, the neon will start to glow and it gives off light. But it's not giving off white light. It's giving off this very characteristic red-orange light that you've seen in neon lights. If you take that orange light and pass it through a prism to separate out the colors, you'll discover that a bunch of colors are missing. That's what's shown in the diagram on the left here. The light from that neon lamp, which is shown on the far left edge of the picture, is being spread out through a prism, and all sorts of colors are missing. This type of spectrum is referred to as a line spectrum or a discrete spectrum. Now, that emission spectrum from the previous slide is due to neon emitting that light. It's converting the electrical energy into light energy, but it doesn't emit every possible color, just certain ones. Well, under a different set of circumstances, we can also get samples to absorb light. You'll have, just schematically speaking, you'll have a light source, and you pass that light through your sample, and then you look at what light gets passed through versus what light was absorbed. You can use a prism to spread the light out into its component colors, or a diffraction grating. You can see an example of a diffraction grating if you pick up a CD and you look at the back side of the CD and you see that rainbow appearance. The CD is acting as a diffraction grating when it does that. And then that light gets sent down to some kind of a detector, which is pretty much like a camera. It's just something that's sensitive to all of that light. Now, the light source will be a continuous spectrum. It'll have all the colors of the rainbow. But what we'll find if we look at the light that comes all the way through into the detector is we will find that some of that light has been absorbed by the sample, and so we now have these lines, these black lines that are referred to as absorption lines, where the light was absorbed by the sample. And for any given substance, if you look at those absorption lines and you compare it to the emission lines for the sample, you'll find that they line up. So in the one type of experiment, we can get a sample to emit light of certain colors, and in the other experimental setup, we can get it to absorb light that's those exact same colors. Coincidence? I think not. The experiment described on the last slide is a rough outline of spectroscopy. Spectroscopy is an experimental technique that takes some form of electromagnetic radiation, illuminates a sample with it, and then observes what part of that radiation was absorbed and what part of that radiation was not absorbed. By looking at different portions of the electromagnetic spectrum, we get different information about our sample. If we are using microwaves to illuminate the sample, we get information about the molecular structure. What are the links between two atoms that are bonded together in that sample? And what are the angles of the bonds that are formed in the molecules? If we're looking at the infrared portion, we will get information about the bond vibrations, and that can also correlate to structural information. But if the light that we are investigating our sample with falls into the ultraviolet or the visible portion of the spectrum, we get information about the energy levels of the electrons that are in that particular sample. So to wrap up with our objectives, light is a portion of the electromagnetic spectrum. Continuous spectra have all the colors of the rainbow, whereas line spectra have only certain discrete, separated lines or colors. And then a sample of matter under one set of conditions might emit light in a certain line spectrum, but under a different set of conditions it might absorb light, and the colors which it either emits or absorbs will be the same because they are due to the arrangement of the electrons in that sample.